Continuing on with our discussion on biological membranes, we will now be discussing about membrane proteins. So key concepts in this part of the lecture is that we have basically three types of membrane proteins. So the first one are the integral membrane proteins. They have transmembrane structures that are basically interspersed, or they integrate into the membrane, hence the term integral membrane. And then we have the lipid link proteins, whereas instead of having hydrophobic parts or hydrophobic domains like the integral membrane proteins, these proteins are covalently attached to the lipids. So this attachment, although it's covalently bound, so it depends on the type of attachments. For example, prenyl attachment and glycosyl phosphatidyl inositol attachments, they are more often permanent attachments. Whereas if it's a palmitic acid or the fatty acid group attachments, there are types of lipid link proteins where there is a reversible attachment. So you have esterases that can detach these lipid link proteins, and this detachment is important as part of a signal transduction mechanisms of the cell. And then the last type of membrane proteins we have are the peripheral membrane proteins. These peripheral membrane proteins, they interact non-covalently with the proteins or the lipids at the membrane surface. So unlike the integral where they integrate themselves, nor the lipid link proteins where they are attached to the lipids, the peripheral membrane proteins are just simply there, interacting non-covalently. There's no covalent bond per se, but they are interacting with mostly the polar, usually hydrogen bonding with the polar heads of the lipids. So having a closer look, so these are the integral membrane proteins. So this is HIV protease mutant, although this is from a PDB RSSP file, but the ritonavir is not exactly seen here. But here you will see this is an integral membrane protein. The green one, these are your phospholipid bilayer. So this is the phospholipids. The green one are the carbon of the fatty acid chains. And then here is um, the surface electrostatic surface potential of your integral membrane. And as you can see, the whites, or rather the gray areas, they are neutral, and these are the hydrophobic regions. And then the most of the hydrophilic regions basically have the blues and the reds, so basically the basic and the acidic regions. Here, negatively charged or positively charged regions, or the electrostatic surface potential is positive or negative. They're mostly concentrated here and here, where it is interacting with the polar heads. And the interior of the bilayer are mostly gray areas. It's neutral charge. They are hydrophobic. And that is how integral membrane proteins become associated with the membrane, because they integrate themselves within the membrane itself. So here is another view of another protein. So some integral membrane proteins have large domains on either side of the membrane. So here is the extracellular side, and this is the intracellular side. And connecting this, basically this type of protein has basically three domains. The domain that is interacting with the extracellular side, again, the outside side. So usually they are signaling proteins or receptor proteins, and then integrating them to the membrane is the nonpolar domain, where if you look at this one, they are mostly nonpolar residues. And then you have another polar domain that this time it's interacting with your interior, or the cell interior in the cytoplasm side. So looking at this transmembrane domain, so this is basically glycophorin A, this type of integral membrane protein, glycophorin A. Now this one, as you can see, it has a domain. This is the domain that is interacting. So if you look at the chain, the protein chain of this protein, you will see one hydrophobic uh, one domain, one long stretch of um, amino acids that are hydrophobic. And so this is basically some in proteomics, we can identify, or we can detect whether A protein is a transmembrane or a membrane protein based on what you call this hydropathy index. This is a hydropathy index. You will see here the amino acid residues which are hydrophobic here on the top and the hydrophilic ones. So if you see domains, stretches of amino acids that are hydrophobic, more often than not, they are most likely associated with membranes. Now, this is another transmembrane proteins that has 7 domains that are interacting with the membrane. So this is a bacteriorhodopsin. So sometimes it doesn't have to be only one. So sometimes they go back and forth. So it has a total of 7 domains. So here is your bacteriorhodopsin. As you can see, the 7 domains that are interacting here, color-coded for your convenience, these are the alpha helices. These are the ones in the membrane. So they, for example, the lipid in this region here is the lipid bilayer, and then these are the outside or the where the heads of the polar heads are interacting. So this is the exterior of the membrane, and this side is interacting with the bilayer. So here is another, this is actually a porin, OMPF, a type of protein that is basically, as its name suggests, it forms somewhat like a pore in the membrane. So it's a porin, it's a transport protein. So materials can be transported across the membrane by passing through the interior of the barrel. So here's another protein. As you can see, not it the transmembrane domains are not only limited to alpha helices. They can also be beta sheet formations that integrate with the membrane. Now here is OMPF. This is actually actually trimeric parts. So each individual barrel, from what you can see from the previous slide, so that is the individual barrel. This is the individual barrel, and 3 of them interact. So this is the top view of this barrel structure. So it allows passage of water usually through the membrane. Usually are porins or aquaporins. It allows passage of matter. Now, for the structure, or how do they in terms of amino acid distribution, if you remember the chain, the backbone of your amino acids are basically N C double bond O C, NCCs. Now they are inherently hydrophilic. The oxygens and the nitrogens are electronegative molecules, and they're hydrophilic. Now these structures, the formation of the beta barrels and the formation of the alpha helices, stabilizes these hydrophilic backbones, whereas they are locked into one another, and then on the outside, because again the ribbon view only allows us to view the alpha helix, or rather the backbone, the protein's amino acid backbone, rather than the side chains. So basically, in transmembrane or integral membrane proteins, the transmembrane portions or the transmembrane domains, the side chains of these domains, they are oriented outside, and these side chains are mostly hydrophobic. So it basically protects this backbone, the hydrophilic backbone, from the interaction with the hydrophobic tails of your phospholipids. So the same with this one. So you have here hydrophobic residues located on the outside interacting with the phospholipid bilayer. So here is another view of this one. So again, the greens are your phospholipids, and as you can see, interacting with them are the grays of the protein. So these are the nonpolar side chains of your protein. So they are well integrated into the membrane. So another attachments are the lipid link proteins. So usually the lipid link proteins, they are attached to the lipid through a modified cysteine residues on the N-terminal side. So here is one example of a sulfide interaction. So this is your cysteine. So cysteine is the amino acid that contains sulfur, and the sulfur will then be attached to your phospholipids. So that's how they are anchored. So here is another example. So this is phosphoethanolamine, and this is your phospholipid, and basically the protein is attached to your uh, protein as anchors. So these are the GPI linked protein. So moving on, so those are the three attachments basically. I won't be showing the peripheral proteins because they are not really attached, but they are just associated in the membrane. So uh, they are usually found floating near your membranes. So moving on, let's look at the structure and assembly of the membrane. So key concepts from this part of the this part of the lecture, that we have the dynamic arrangement and interaction of membrane lipids and proteins are described by what you call the fluid mosaic model. And since the membrane is very fluid, it must also have strength. And so we have membrane skeletons that gives the cell specific cell shape. And yet, because of the fluidity, it becomes more flexible. So the skeleton gives it uh, enough shape as well as flexibility of moving, especially seen in erythrocytes, basically red blood cells, where they must be, they have this characteristic flat donut shape, and yet they can become flexible so that they can um, be able to pass through the very, very narrow capillaries. So lipids are not distributed uniformly throughout the membrane, and they also form raft, and we call them the lipid rafts. So the secretory pathway of membranes, which describes the transmembrane passage of membranes and secreted proteins. So this is in the formation of the membrane. So we have also different types of coated vesicles facilitating these transports. So, but in this part of the lecture, we will only be focusing more on the fluid mosaic model and your membrane skeleton. So here is your plasma membrane showing you your the different membrane proteins. Although we say we focus more on the membrane proteins, that doesn't mean to say that's only the proteins interact with our phospholipid bilayer. There are also oligosaccharides, the carbohydrates, carbohydrate groups that are interacting with the membrane. And you mostly see carbohydrate groups on the exterior surface or the exterior side of the membrane, because most of these glycolipids, these oligosaccharides, have roles in cellular signaling. In fact, our ABO blood type system is not the differences between blood types A, B, and O are not due to proteins, but rather through the type of oligosaccharides that are in the exterior of our red blood cells. So basically, you have here your plasma membrane and the fluid mosaic model. It shows us that the plasma membrane, as we can see, they are not this bilayer. They are not covalently linked with one another. They are freely moving, and so they are like uh, a sea of lipids, and then you have these integral proteins that are floating and moving around that sea of lipids. Um, they can be they can be just floating at the top or at the surface, just like the peripheral proteins, or they can be just like the iceberg. So you have a tip at the top and a large submerged part at the interior of your membrane. They can also be linked to your lipids itself. So those are basically the well, you can, if it's a fluid or rather fluid mosaic model, so you have a mosaic of proteins floating around a sea of lipids, and that is your uh, fluid mosaic model in a nutshell. So how do they know that they are moving, and how do they know that it's like a fluid mosaic model? So basically, this is through a an experiment in immunofluorescence experiment. So where they fuse a cell of a mouse and a cell of a human. So membrane fusion is possible because again, there are lipids. The lipids interact with other lipids, and so they can be able to fuse two types of cell, two different types. So you have a mouse and a human cell. So upon the fusion of the mouse and human cell, after some time, they observe that the distribution of the animal, which is the mouse proteins, and the human proteins becomes mixed or interspersed within the cell. So the green here shows the mouse proteins floating around the membrane, and the red is the human proteins floating around the membrane. So when they fuse the two cells, after some time, they can see that the proteins move around and become more interspersed. And that is one of the foundations of the fluid mosaic model. So meaning to say, our cell membranes, our biological membranes are very dynamic structures. So another evidence of this fluidity of the membrane, the movement of the different structures in membrane, is the FRAP measurement, or the fluorescence recovery after photobleaching. So basically, you have here a laser that bleaches your cell. So basically, you have a cell incubated in a fluorescent mark, and then when you focus a laser there, it becomes bleached. So this white spot on the cell, after some time, will be gone. So if you look at the structure or the signal of the fluorescence intensity, so because it becomes bleach, the fluorescence intensity goes down, and then after some time it recovers. So that means the part where it was bleached becomes interspersed with the part that is not. So they can become mixed up, and hence you have your recovery period. So it can also be used to measure how fluid the membrane is because of the recovery time. So although the membrane is fluid, because of course, fluid structures have also its own dangers and disadvantages. You cannot There's a difficulty in holding its shape, and that is why we have proteins called the membrane skeletons. They are proteins found in what they call they were first discovered in ghost erythrocytes. Basically, these are erythrocytes that have been lysed before, but because the membrane can self-seal itself, they reform again. Although the interior is basically empty of the contents of the normal red blood cell, but the membrane itself becomes intact, or rather it reforms again, and hence they call it the ghost red blood cells or ghost erythrocytes, because they are technically dead cells, but their membrane becomes reformed. Okay, and one of the proteins found them is spectrine. Actually, the naming comes from specter, which is another term for ghost, because again, they're found in the ghost erythrocytes. But anyway, this membrane skeleton, spectrines, they help stabilize, or they help in keeping the membrane, although it is fluid, it helps in the membrane, the cell maintaining its shape. So how it does is because of the structure of this spectrine. So you have here repeating alpha helix alpha chains that are arranged in a triple helix arrangement, and there are uh, two two subunits for this spectrine. So they are arranged anti-parallel with one another, so N to C, and then again the other one is C to N. Now I said they are stretchable because they're like garters. So these alpha helices, they can be stretched depending on the stress that are applied to them. So here is another another look. So they are polymeric. The spectrin is polymeric. They are polymeric proteins. You have repeating sets of three repeating subdomains. So that are each repeating unit is actually arranged in a triple helix formation, which can be stretched out. So this makes spectrin somewhat like a garter, so it can be stretched. So here is an electron micrograph of the surface of your erythrocytes showing these spectrines. So these are the membrane cytoskeletal elements that are associated with this, basically this membrane. So as you can see, it holds the shape of a membrane. So that's one of the ways on how, despite being a very fluid structure, the cell membrane, our cells can still retain their characteristic shapes. For example, the shape of the red blood cells, which is somewhat like a flat donut, and even the shapes of your neurons. So you have these characteristic star-like shapes. So although the membrane is fluid, they can hold its shape, and they are also flexible enough for the cells to adapt. So here is the diagram of that part. So if we look closely at this segment of the cell, the membrane of your erythrocyte, we will see here these spectrines crisscrossing around this structure. So that allows it actually serves as support to hold the shape of the cell together. Okay, so aside from that, we also have ankyrin. It helps in the anchorage of your membrane cytoskeletons. And then, as you can see again, you have um, hook-like membrane structures on this one. So the mobility of the integral membrane proteins is actually affected by this spectrin. So as you can see in the previous structure here, because the spectrins crisscross around your on the membrane, of course they actually form fences, and it actually affects the movement of your integral membrane protein. So if they become fenced on one side, again they won't be able to move around much. And if they you have pathways which they can move around, or in some cases some integral membrane proteins are actually associated, they serve as anchors uh, to the spectrines, to the membrane cytoskeleton, and they become held in place. So these membrane cytoskeletal elements actually also keeps it regulates the movement of the proteins, the membrane proteins in your cell, the cell surfaces. Now, aside from those movement, you have also what you call asymmetric arrangement in your phospholipid distribution. So again, we have, because it's a bilayer, you have an inner and an outer side. So two layers, an inner leaflet, we call it a leaflet, just like, say, a book. The inner and the outer leaflet of your phospholipids. And it was observed that there are some asymmetricity in our there's an observed asymmetry in the type of phospholipids that are found in the inner versus the outer lipids. So here is in terms of percentages. So sphingomyelins and phosphatidylcholine are usually found in the outer leaflets, whereas you have phosphatidylethanolamine mostly on the inner one, and phosphatidylserine is almost always on the inner side of your membrane. So this type of movement is driven by two factors. First is the concentration gradient, of course, and this is the spontaneous movement or flip-flopping. I already discussed that one in the previous part of the lecture, in the previous video. So these are facilitated by flipases, and they are facilitated diffusion. So it's a passive type of transport. Now, because we have observed that we have here asymmetry, and the equilibrium doesn't like asymmetry. So that means there is another driving force behind this asymmetry, and these are the active transport. Basically, it's ATP driven. We know once ATP becomes involved, this becomes an active transport. And these are the phospholipid translocases. They transport certain types of phospholipids against the concentration gradient. And that is how the cell membrane maintains its asymmetry. Some types of membranes are more often than not found in the outer leaflet, whereas others are only found on the inner leaflet. Okay, and that's it for the membrane structures. In the next part of the lecture, we will talk about transport across the cell membrane.